What's up, everybody? Welcome to the first actual live broadcast here on Logical, Plausible, Probable. I am here with the one and only Sal Cordova, and we're going to be discussing a variety of things ranging from protein synthesis, structural biology, the quaternary aspects of the whole protein process, and also going to kind of touch on how all of this kind of works directly against the common argument by the folks that I personally go up against on a regular basis who just like to dismiss all of this with, hey, it's just chemical reactions, y'all. It's nothing beyond that. There's no additional variables and things that are required well prior to any of the cascade effect chemical reactions that they love to rely upon. So Sal, thanks so much for joining us. And just so everybody knows, this man has, I think, 5 different degrees and has worked on things in information technology through engineering. He's invented and built things that are now on aircraft carriers, and has actually spent the last 6 years doing direct research on this topic of protein synthesis, especially with a very hyper focus in human proteins, and doing this directly for the man who actually invented what's called the gene gun. So basically anything you have ever run into, eaten, or experienced in the context of genetically modified anything, Sal actually works directly for the man who invented that. So Sal, thanks so much for taking the time to come on and join us and share your knowledge and the research you've been diving into for the last 6 years.
Oh, thank you for having me. And I guess the theme of the show is to try to show that DNA is not like Diet Coke, and that it is chemicals, but they are organized chemicals. They are chemicals that are not the expected result of random, either random mutations combined with natural selection. And that's what I hope to discuss today. Both will fail. They'll definitely fail in the scenario of abiogenesis, where there's really no natural selection, which we would call chemical evolution. And it will fail even in biological evolution, which is generally known as evolutionary theory. So I'm going to cover some of the chemistry behind that and why actually physics and chemistry it makes it possible, but also improbable. That's a principle that I've tried to convey to people. The way we detect design is that the design is physically and chemically possible, but it is also simultaneously improbable. The way that we know something is designed is that obviously when it's sitting right in front of us, we know that it exists. It's possible to be constructed that way, but it's also highly unlikely. It's just like a house of cards or a configuration of coins that are fair, and you have 1,000 of them, they're all heads. It's that configuration is possible, but it is also improbable. So we'll cover some of that today.
Quick interjection on that, because one of the very common supposed counterpoints about that is, they leverage deep time and large numbers and all these different things to try and somehow account for what you just talked about there. And I think very often the layperson kind of misses the point of that you're making in line with all that of, it doesn't matter if you have a 6 trillion headed dice. It's still not a remotely conceivable premise that all of these things will happen in the appropriate order, which is something that we're going to talk about, right? All of these things have to happen not one at a time, but simultaneously. Is that a fair point to make?
Yes, that's actually a very fair point. And there are some people that will say, like Robert Shapiro, they'll admit, they'll concede that it looks really unlikely. Even, oh my goodness, Richard Dawkins. Richard Dawkins will admit that the origin of life seems a remarkably lucky event. So the question is, and I pose this to people, I mean, just let people try to be as honest with themselves as they're willing to be. And some are quite open about it. I say, well, how improbable does something have to be before you'll begin entertaining a different explanation than the natural ones, or you'd be willing to say, yes, this is a miracle. At least you might deny that God had anything to do with it, but when will you classify it as a miracle? If their answer is there's no level of improbability, then it's like, well, okay, then nothing will convince you. I offered this straight up to Tracy Harris on the Atheist Experience show sometime in December, just before Christmas, 2018. I covered the passage in John where Jesus heals the blind man, and then I related the story in the modern day where astronaut Charles Duke, who was the youngest man to ever walk on the moon, he was not a Christian at the time when he walked on the moon. He came back, his life was a mess. His wife wanted to commit suicide. His marriage was on the brink, and he was becoming an alcoholic. I mean, he was just, he made it to the moon and he just came crashing back. In that process, he became a Christian. Sometime thereafter, he led a prayer meeting, and there was a blind girl there, and he prayed for her, and she got healed almost instantly within a matter of minutes. So I have no reason to doubt him. I mean, he was a national hero. He had it all. He's not like some people that are just, one would probably reasonably suspect they're just making up stories to get attention. That would not be the case with him. And the way he's lived his life, now later in life, he's spending his time reaching to the outcasts of society, visiting prisons. That's characteristic of someone who wouldn't be trying to just make up stories to make headlines. And so I consider that valid. So I related that story to Tracy Harris. I said, if I were that blind girl, and by way of implication, if I were that blind man that Jesus healed, I would follow Jesus the rest of my life. And I said, well, what would you do? She basically said she wouldn't. And she said, well, I would try to figure out a naturalistic cause. And maybe we could just find out how it happened and we could heal everyone. And I mean, there's merits to this in, one way or the other. But I told, when I heard that, I said, yeah, definitely the path that she's chosen is not for me. Because for someone like that, no amount of miracles would be enough. Right. So that's the philosophical side. All that we can do is demonstrate that at least as far as we know in the observable universe, certain structures, certain features are beyond the reach of reasonable chance. When it's so astronomical, it's way more than all the, not just the atoms, but all the possible interactions. It starts to become absurd.
Something in that context of the improbability you're talking about, I find very interesting, is that in basically any other discipline other than origin of life / evolutionary biology, probability is a direct, correct me if I'm wrong on this, but it's considered a direct component of whether or not you're even going to go down a potential and try and investigate a potential hypothesis. If the improbability is above X, then it's we're not going to waste time on this because this is just completely ludicrous. Is that a fair position to say about pretty much every other discipline? And from things that I've read, I'd love your opinion on what I'm about to say here. Things that I've read, the standard for improbability is, in context of something they would not pursue, is so comparative to the admitted requirements for origin of life and then evolution as an extension, aren't even in the same stratosphere. I mean, they would dismiss something not even before you can get a fraction of the way to where the improbabilities are for origin of life. Is that a fair position?
I think that's fair, and probably the fairest thing is just to ask. Some people will be candid enough and say, well, I don't care what the odds are. We're not going to accept design or some sort of conscious intelligence as the explanation. And that's basically what one of the top evolutionary biologists, Eugene Koonin, had said in his Logic of Chance and other papers he published, like on the eternal inflation. And even though Kunin specializes in evolutionary biology. He has dabbled into abiogenesis, which I think it's fair to say it really is a different phase. If you think of molecules to man, you might say there are two phases. So I'm not going to argue whether one's the part of the other. That's just however way one wishes to split the hairs, it's still a problem. Abiogenesis will always be a problem. But anyway, he dabbled into abiogenesis, and he was so unpersuaded by the various arguments, the chemical experiments that have been put forward. He postulated that there are infinite numbers of universes that we can't see, we can't test, we can't know. We just infer by maybe not a very good line of inference if one accepts certain parameters of the Big Bang, which is also doubtful. But the point is there's no way he can prove it. There's no way he could experiment, no way he could test. Everything that some of these atheistic people would accuse creationists of appealing to, he's appealing to, except It's just not a conscious intelligence. And so he appealed to multiple universes as an explanation for the origin of life. He said that's the proof. It almost seemed, I mean, these are not his words, but the impression one could get from reading what he said was that the proof that there are other universes, that they have to exist, is the fact that the origin of life is so improbable. So that means that's kind of his logical inference, because that's the only way you could explain it. The alternative is intelligent design, and he just dismissed intelligent design. He said, look, all these multiple universes are such a better explanation. He specifically said no need for intelligent design, and I was just like beside myself.
Well, and what's very interesting about Kunin, especially given his significant role in biosemiotics, and I've read several of his papers in the context of genetic code, and it's very fascinating to expand on what you were saying about his dismissal. Is in his papers he freely admits the conundrum of the genetic code itself, and which I find very fascinating when I listen to the standard atheist talking points about the genetic code. And I'm like, a guy who's literally an atheist, who depends on and argues for the multiverse in order to explain the improbability of this, also freely admits that the genetic code literally exists and that we have no explanation for it. And to me, when you think about that, it's at what point do you have to concede that the folks on the other side have absolutely no clue, while simultaneously claiming that we are clinging to a God of the gaps mindset, when per their own admission, they don't know how to explain the variables that directly point to an intelligent agent being required. I mean, I find it just fantastically ironic, that whole back and forth dialogue. And I'm like, staunch atheists agree that this is a unavoidable argument, and you guys want to just dismiss it out of hand when they admit that it's the unanswered question.
I thoroughly agree. And the reason I frame that is, okay, so we're not going to agree on what will ever persuade someone like that is absolutely true. And that's fine. It's just like, let's get that out of the way. It's like, I know you're not going to be convinced with any argument I put forward. So what's the next thing we can agree upon? Can we at least agree that something's improbable? And obviously, Kunin and I could actually agree that certain things are improbable. Ironically, Fred Hoyle, who used the word intelligent design, was an atheist or agnostic. There's probably some subtle differences that are not really worth arguing over. He was basically not a creationist, but he advocated some sort of intelligent design because he saw the improbabilities. Of course, he appealed to things like space aliens, and he was the originator of the spaghetti monster, wasn't he? I don't I don't think so, but if he is, so
I think he made that comment as a joke, and then it was I think it was expanded upon by Dawkins, and then it's been just absolutely seized by modern atheists. But so in line with everything we're talking about on the improbability aspects, let's talk about something I know you're very passionate about, which is the whole element of structural biology.
Yes.
And
okay, so
because I think that's kind of
the
foundation of where we're going with this conversation. Would you agree with that?
Yes. If you see me looking left and right, I have like 4 screens because as we're talking, I'm trying to prepare little slides for our viewers. So I'm not trying to be disrespectful or anything to you.
Oh, no,
when you're
I
talking to me.
I figured that's exactly what you were doing. So kind of give us
and I'm actually even looking here. There's some people. I'm even looking at our readers. So let me go into some probabilities. And I think the reason I want to cover certain things is the reason I like structural biology. Rather than what we used to do, what creationists used to do is they would quote studies about the improbability of proteins. But now with the advent of structural biology, we've been able to get physical models of how these things look. With just a little bit of training, one can actually see why it's improbable. For example, if you saw a nut in a bolt, something as simple as a nut in a bolt, you would know that it's not likely the product of random chance that could shape the two that could perfectly fit. Well, a lot of structural biology arguments that I use are actually just that. Just think. Because rather than just quoting some figure of how astronomically remote this is, I can show pictures as to why. So
you want to go ahead and share your screen so we can
yeah, I'm going to do that now, and this is a protein you're familiar with. Share screen. And I can actually see, I have a little monitor. It's a little bit delayed, but there it is. It's the homohexameric helicase. So you're familiar with the helicase. Why don't you tell the audience what the helicase molecule does?
So correct me if I'm...
Speed of sound.
I think I lost you there for a second. Sal, were you asking me to explain?
We're back online.
Okay. So you're saying to explain the helicase a little bit?
Yep. Okay. If we had a video of it, if you want to try to grab a video of it and show it, it's only like a minute, or I could try. Ooh, sorry. I can try to get it. Later when I unshare my screen. But go ahead.
Yeah. So for this is a very significant component of DNA transcription, and I find it very interesting how in my debates on a regular basis and in the comments afterwards, when I have made this argument of, hey, there's a literal nanomachine that is required for something as fundamental as DNA transcription, people dismiss it and just say, oh, it's just a chemical reaction. Well, not only are there, outside of the helicase, correct me if I'm wrong on this, Sal, but there's quite a few other components that are required in addition to the helicase. This protein literally unzips the DNA double helix and does it at the speed of a jet engine. And we're talking about a physical process of unwrapping that enables the whole transcription process to exist. Is that a fair assessment, Sal?
Yes. And actually what I'm going to do, I'm going to try to multitask here, which I'm terrible at, but I'm going to try to do it anyway. Maybe when you talk, what I'll do is I'll try to bring up videos of this because that'll give the wow factor. So I'm going to get the boring part of this. Since you've seen this, I'm going to discuss what they call quaternary structure, and you'll actually see why this is highly improbable. If you see the different colors here that compose the helicase, that is actually what they call the helicase protein. So each one of these that's a separate color, they're actually sequentially identical. You take one gene and you make 6 copies of it. One gene, you make 6 copies of it, and then the cell sticks them together. So this again is a little bit like nuts and bolts, but you could see how, if you could see my cursor, you could see how it has to just have, now again, this doesn't, at the molecular level, it doesn't exactly work like nuts and bolts at we call the macro level, at human perception. At the molecular, things connect. It looks kind of wiry, but they are very careful. They have to be positioned right so that one piece will bond to the other piece in just the right way and just the right orientation. So one can imagine if the sequence, if the spelling of the protein were different, it would be like a password. So think about this as like a password. The spelling of the protein in this part of the protein has to complement the spelling of the protein in that part. But remember, this is the same sequence. This is the same polypeptide sequence. I'm sorry if I'm losing some of the audience because I'm using some technical terms, but people who are biochemists would totally understand this. And I could show you the spelling of the helicase, but the idea is the way that the whole thing is spelled, because the spelling of the protein is in terms of amino acid sequences. This is like a paragraph of about 1,200 letters or so. I don't know off the top of my head how many letters it's composed of, but so just imagine that some of the letters in that sequence have to be able to be, there needs to be foresight that how this was put together was that, let's say hypothetically you or I were trying to design this. What we wanted to do is design a hole just this big, which is where the DNA single strand will slip through. It's preferable that it's not too big. And you're going to spell the protein in such a way that when you throw 6 copies of it, it's going to get assembled into something like that. The probability of that is extremely remote. And plus there are other things. Because if you can imagine it, it'd be very undesirable if these things were sticking to the wrong thing. It could just be, it's just like taking pieces of tape and just randomly putting them together. You'll just get a mess. But the way these work is that they do adhere to each other, but they will adhere to each other in a very specific way. And amazingly, in many cases, for other proteins, they'll spontaneously adhere in just the right way. This would be like taking pieces of tape that are just matched for each other, throwing them in a pile, and they're going to find each other and snap together spontaneously. There is some degree of self-assembly. Now, in the case of helicase, there are 2 machines involved with the helicase. One is called the loader. It will actually assemble this around the DNA, but this won't. It can't be force fit. It has to be. There has to be foresight in this design. So if we were putting this together, we would have to be very methodical. The thing that we're learning is, and I mentioned this in my Sunday night debate, May 31st, with Ron Garrett on genetic entropy, that this is the hard protein folding problem. The hard protein folding problem is to say, let's try to make a device that has these requirements. Now create a sequence that will fold and make the machines we need. We don't know how to do that. We don't know. We can only tweak. And when the person I worked for, yes, please.
Quick interjection on that. In the context of, I mean, I know that we're attempting to do synthetic protein synthesis, right? And I was actually watching a interview with several synthetic chemists who were working on this whole thing, and it was very interesting to me listening to them talk about how they were almost completely reliant on, or becoming reliant on, supercomputers, number one, and number two, they kept commenting about how vastly superior Mother Nature was in relation to the proteins that already exist versus what they were being able to create. And I thought that was a very interesting concept to consider, especially when you think about this from the improbability perspective of, okay, humans are using supercomputers to fail miserably at creating anything equal to what is, the fundamentals that are required for life. And yet we want to completely dismiss the premise of, hey, we know there is no less than, I think, what's the current standard? Like 250 proteins are required for the simplest form of life. And
400.
Oh, it's 400?
And that's not a very good, that life would actually have to depend on it presumes the existence of other life because it's a parasite. There's lots of things that can't, lots of nutrients that can't synthesize from scratch. So
you're looking at, so I'm guessing, is this the spelling of the helicase?
This is the spelling of, okay, so let me try to explain for the readers. This here, when I was talking about the spelling of a protein, this is what the helicase if we were to spell this out and get a cell to print it out, the ribosome to print it out. So just say that you have, this is kind of the blueprint here. The DNA will generate it has an equivalent blueprint in its language, but then it will translate to this language, which is represented by English alphabetic characters. So this M is an amino acid. The G, I hope you could see my cursor, is an amino acid. So M is methionine, G is glycine, D I think is aspartic acid, V is valine, K is lysine, N is asparagine, F is phenylalanine, L is leucine, Y is tyrosine, et cetera, et cetera. I may have gotten one of these wrong. So there are 20 canonical amino acids, and this is how you spell the helicase. And let me see, does it give me I don't think I have it's kind of unfortunate. Let me see, just give me 1 second. See if it gives a number of how many.
Something else I think people need to recognize is that what you're representing there in terms of spelling via representation of the individual amino acids, the genetic sequence which was translated into that is literally 3 times as number of factors required in terms of actually more than that when you think about it from a base pair sequence perspective. But it requires a triplet of base pairs to equal 1 codon, which then equals 1 amino acid. So when you really think about the chemical construction of the instructions for the blueprint for the spelling of this protein that we're referring to, and Sal's explaining here, you're going to whole new levels of, you're adding additional layers of organization that are required. Is that a fair
oh yes, absolutely. We're not even scratching the surface. So I apologize to the viewers here for the screen jumping around. We're kind of doing this on the fly, which is okay, because I learned too how to better communicate what I'm trying to say through the process of interacting, and then I might be able to make slightly more formal and structured lessons in the future. But so the point, John is exactly correct that it'd be 3 times more DNA. So what I was trying to do just now, I'm trying to see how many, I'm trying to give you a hard number on how many of these. Can you give me a moment here? And
yeah, no,
if you wanted to say something while I look this up.
I'm happy to talk about this. I mean, I think something that people very often forget about when they're, considering A, how does life exist? B, what would be required for, just undirected mutations to result in a new functional protein. Very often the kind of the foundational, improbability arguments are, looking at, somewhere between 100 to 150, base pairs being required to equal this simple protein. What people forget is, and I'm going to wager that the helicase is probably a couple thousand amino acids base pairs required for those amino acids.
No, I'll give you the figure. I found it 1,270 amino acids. So you multiply that by 3. That's almost 4,000 nucleotides,
Right.
4,000 bases.
Exactly. And to me,
Big.
and when you think about this,
4,000.
and I know you're going to talk, I imagine you're going to talk about this later in terms of how correct the spelling has to be in order for these machines to work, is that we're not talking about being able to just have random misspellings until you get to the right one. The machines don't work if they're not spelled correctly.
And in the case of helicase, life doesn't work. The thing will be dead.
Right. So you have to have the equivalent of, let's see, you're probably looking at like, I'd say no less than 10 pages of correct language instructions with 0 errors in order for one of the absolutely most fundamental, and correct me if I'm wrong on this, but I don't think that even the atheist evolutionary biologists will argue that without helicase, life cannot exist. Is that a...
I don't know of any. Now, what I try to get around is, let's just say hypothetically that we could construct life differently. The question is, could the life that we have exist without it? This is like saying, okay, there are a million ways to make airplanes, and they'll say, well, you're just picking that one, and you're fitting the probabilities. But the way this should be described is, well, no, you're looking at it wrong. The way you calculate the probabilities is how the parts connect, because you can't take the parts of, say, a soft-width Camel, which is a World War I airplane, and fit them into a 747 or vice versa. So the probability should be calculated in terms of connectivity, just like nuts and bolts. That's why I love structural biology. You could see it. So even though, and actually there are different life forms with slightly different genetic codes, and there are definitely different architectures, like say between prokaryotes and eukaryotes. So we can implement life different ways, but the question is, what's the probability that the structures connect in a way that is life critical? That's the better way to frame the argument. So right now we live in life that requires helicase, and I have a video that I'll show shortly that'll show the complexity. So while you were talking, I was ginning it up. So please go on, John.
Oh no, I mean I guess just the point I agree with everything you just said there, and I think it's this is why I very often try to relay to people that are not, or kind of refusing to even consider the arguments of, you have to look at what is rational in regards to the point you made of, this is how it is, not how it could be, it's how it is. And in any other premise of deduction, induction, forensic analysis, whatever it is, you would apply only the how did this occur to what you observe. And it's like in the context of our very existence, which in my opinion would probably be the single greatest question that can ever be asked, is, oh no, let's just completely toss out the parameters we would use in any other context in regards to the most fundamental question that can ever be asked.
And maybe another way to frame it is, this is what I use as an analogy. There are infinite numbers of making a lock and key system, right? I mean, there are a bazillion ways you could form lock and key. But when we encounter a lock that perfectly matches a key, and it's a relatively complex, we're not talking some trivial key or lock system, something with lots of tumblers, and then the key would have to have lots of notches. If we see those things match, we would not think that someone was just kind of, taking a hammer and chisel and just randomly chipping out the key. So that's the connections we see in life. We see parts that are well fitted to each other. And I know this looks kind of wiry, but for anyone who's a biochemist or structural biologist, they will wonder at how this connects, because it has to be so precise. This spelling has to be just right. So like I said, So if you look at each of these colored strings here, we can call them polypeptides. We can call them individual proteins. To make a functioning helicase, we take 6 copies of this, we print it out, and it will naturally fold in shape, just like lock and keys, and they'll hook together like that. That's only the first problem. This is why I was trying to point this out in my debate on Sunday night, May 31st, 2020. I said, this is so tightly constrained to work as a functioning whole. At best, changes to this, you would hope that they're neutral, because the only way that changes will affect this is for the bad. It's either neutral or bad. It's not, you're not going to improve this. It's just like trying to improve the connection of a nut and a bolt. I mean, how much more can you improve it? If you make the bolt too large, it's not going to fit. If it's too small, it's too loose. It has to fit just right. And that's why I wanted to get on evolutionists and say, you can't be, they'll be arguing about distributions. I said, you got to go to the molecular biology. It will not be as forgiving as what you're saying.
Right.
And the other thing that I came out with, and I came out swinging, I said, evolutionary biologists are so bad and so desperate that they'll redefine the meaning of what fit means. To them, fit means, oh, we can reproduce more of this if we knock out that gene. And literally, that's what's been happening. I mean, we have lots of observational evidence that what we call beneficial mutations are actually functional damage. And I said, the fundamental problem, you guys are so desperate, you have to redefine the meaning of beneficial, because you can't define it in terms of structure like I am here.
Well, and I think something else to kind of expand upon the point you're making there is they completely dismiss the requirements of the promoters and the different transcription factors and all these different things. And what really gets me is that they, even though they recognize this, I was reading a paper on this last night of how very often these additional variables that they're saying don't really have any impact, and oftentimes called junk DNA sequences or components and such, is they're a million base pairs away from the gene that's about to be expressed, or a couple thousand. Either way, when you really think about that, you have a completely separate component that is required to execute this gene sequence, which results in the spelling that you're showing us here, which then results in this protein that has all the parts and the components to connect to itself to execute a physical machine process. But all of this happened through undirected process. And to me, when you really think about the arguments that are being made, it kind of blows the mind. Now, what's this video you're showing us here?
I apologize again to the viewers. I've been trying to get a helicase, and then sometimes they just throw random stuff. You know how YouTube can be. So I'm showing the helicase here, and I'm going to start the video. Can you get the sound on here if I turned it up?
We should be able. I think you have to.
Okay, let me restart this. What I'm gonna do is all the boring stuff that I did with the lettering and the quaternary structure, where I'm saying these identical things hooked together. Now you'll see it work as a machine. And what you're saying, just like you said, it's like a jet engine. The speed that this thing works. So here we go.
I think you may have to say like share computer audio. Yeah, I'm not getting the audio from the video.
Really, I'm getting it on the monitor feed.
I think you have to click I think in the share options. I think you have to say
Can we ask the audience? Hey guys, anyone that's watching, were you able to hear this and see it?
Okay, they might be able to, and I'm not. That could be.
Yeah, they are straight crazy. Someone said that. Because I have a second computer here, John. I can see, but we cannot hear the video.
Yep.
Let me try starting it again.
We may have to when you let's see. There's got to be an option for share computer audio.
Okay, I'm sorry. I apologize. Let's try this again. They can see it. What do I have to do to get this to?
I'm thinking, I know with Zoom, there's usually an option for like, you have to checkbox share computer audio. I'm not sure if there's a similar one in.
Okay, I'm going to just narrate this.
Okay, no problem.
All right. So we have this is the DNA here. This is the double helix. And when we're going to try to do replication, we have to kind of pry them apart like that. And we're going to insert a helicase in here. And there'll actually be two helicases, one that goes this way toward the left and one that goes toward the right. So helicases separate nucleic acid duplexes into their component strands using energy from ATP hydrolysis. The crystal structure of this DNA is from bacteriophage 2. It reveals a hexagonal arrangement of 6 identical subunits. Like I said, that's again where you take that spelling that I showed, you just make 6 copies of it. Now, the ring is not actually symmetric. It's slightly squished, and the arrows are showing where it's squished. Now, watch how... So the model will show why there's a structural asymmetry. So these are ATPs here tightly, and these are ADPs, and these are empty regions. So these 3 states interconvert to create a ripple effect that actually makes the helicase turbine turn. So now you'll watch it. See that? Because they're conformational changes, you could see the loops. Because of those conformational changes, this is how it moves. It kind of squeezes together and moves, but then it'll also cause it to rotate. See how it does that? And that's how. So what's happening is as this helicase is moving, it's pulling the DNA there and it's splitting it apart. So what I want to point out is just like in a jet engine, you're constantly feeding it fuel. And these little yellow dots are the ATP that has to be constantly fed. And then that's how this thing works. So I'll tell you what's really amazing. I challenge anyone who doubts the genius of this, try to come up with a system. You'd think it'd be trivial to just write. This is only 1,270 characters. Do you think you could write 1,270 characters to make something like that, a machine like that? It's only 12. We type this much every day if we're arguing on on the internet. Surely it shouldn't be that difficult, should it?
Well, and when you really consider this in the context of the foreknowledge requirement of you have to have a component that is spelled correctly prior to its formation in order to physically have all the appropriate components to interact with a completely separate chemical structure, i.e. the DNA, and have the power component, as you were mentioning, in order to unzip something that is actually is stable, right?
Right.
And I think that's something people forget about from the chemistry perspective is how the helicase works to unzip the DNA is actually rather phenomenal. Is that a correct statement?
It is. And, we underestimate because it's like, when one will look at this and say, oh, that's only 1,270 amino acids, no problem. But the problem comes is that's how we realize how improbable this is. because our best scientists can't explore the space of even a measly 1,270 characters. They cannot find it. So, I mean, you can just imagine this roughly. It's on this order. It's not exactly. It's a little bit less. 20 to the 1,270 power possibilities. So our best scientists cannot, even for a measly sized protein like this, and there are other proteins that are even larger, even this quote unquote simple problem will overwhelm our supercomputers. It'll overwhelm our supercomputers. If we said just design a helicase from scratch, pretend you didn't have an existing model, I mean, this is purely hypothetical. Just say, see if you can make something that will do this, and now spell it out correctly. It can't be done. It can't be done. When you mentioned Dr. John Sanford, who I've had the privilege of working with, he's a genetic engineer. And in his heyday, what genetic engineering meant was not taking DNA and respelling it to make new proteins. No, we took pre-existing proteins. Maybe we stitched a piece here or there. I mean, big maybe. It might be done now. But in his day, what you would do is you take a protein from one creature and put it in another creature. That was genetic engineering. It gave the misleading impression we actually figured out how this works. But to this day, we don't know how to do it as well, as you said, as we can't do it as well as the machines we find in nature. They are so exquisitely put together, and it should humble us. And it should make us think, well, then if it's going to take this long and this much effort to assemble this with all our best minds, why should we expect it to ever assemble by chance? And right. One can work out the probabilities, and the numbers are astronomical if we did by chance. Time will not be adequate. It'd still be a very remote probability. That's why Eugene Koonin was invoking multiple universes. So the mathematicians would recognize this quickly, although, fortunately, the average individual would think it would be enough. It's nowhere near enough.
Speaking of the intricacies of the machines and things, I would love it. I know you have some great slides on like showing the transcription complex and some of the other pieces. I'd love to if you would show some of the things in regards to the folding and how the transcription factories, I believe is the word or the phrase you used.
Oh, okay.
Kind of just and showcase how we're not talking about just, oh, cool, it's purely linear. No, there's actually 3 and 4 dimensional variables that are required for the outcomes that people just take for granted and say can just happen. Because to me, when you really start to factor in, okay, we have a fundamental information source that's required of prescriptive information, not just the general Shannon information definition. We've got formal, functional, prescriptive information that enables in the gene sequence that enables the spelling or translation into the spelling that equals the protein. But then on top of that, you have to factor in physical, spatial temporal factors in order for all of this to be executed. To me, that just takes things to a whole nother level. And I think the average person just doesn't even understand, or I think many people don't even have they don't even know this is reality on how some of these things work.
Agreed. And the people that will learn are the people like those in our chat channel who are taking time to learn this. And the nice thing is, yes, this was technical, but just think, if you're even grasping, if to the listeners out there, if you've even grasped half of this, just ask yourself, if you've not had any organic or biochemistry, look how fast you're learning this. The schools that teach this are designed to train pharmacists and organic chemists and medical doctors. They haven't designed a curriculum where they could try to get you through all this very fast without having to go through 3 years of college, but it can be done. I mean, just think if you could that's why I like structural biology. A lot of this becomes intuitive and you could see the problems if you think about spelling things just right. So as we've been talking, I've been trying to gin up these. Let me just get the PowerPoints I had for my prior discussion, and maybe we can sift through some of them. And I'm sorry if kind of doing this on the fly, but this is also how we learn, how I get to learn what people are interested in.
A quick interjection. So, folks, as you see those little balls that are there's arrows pointing to them, showing histones. When different aspects, and this varies dramatically between cell types, because there's whole components of DNA that aren't necessarily needed in, this section is needed in cell type A, it's not needed in cell type B, or it's only needed in this specific condition. We need this one. So in order to make the expression process much more efficient, it's like zipping of, let's say you have a whole bunch of data on your computer and you say, I don't use this very often, but I might. Therefore, I'm going to put this into a zip folder. And if I need it, I'm going to unzip it. That's literally what we're talking about here. Those histones and the wrapping component, there's many, many uses for this, but I'm saying just from a fundamental standpoint, it's like zipping something on your computer. But something I thought was very interesting is that things like transcription factors, different ones, they literally are able to hop over those histones and then go to the next spot they want to. And we're talking about from a physical hop. So it's shooting along DNA, comes to one of those spots, it jumps over it and goes on to the binding site it's looking for to express a specific gene. And to me, when you think about that, that's kind of that's a mind bender in and of itself.
It's actually more complicated than that. It has to do with this is a picture of chromatin remodeling. It has to actually slide the histones aside, and then the transcription factor can bind. And when I argue chromatin evolution, the debate's over in 30 seconds, because if my counterpart is even halfway savvy in biology, I'll show this diagram and he knows what's coming. I'll say, how did this evolve? Because if it doesn't work, it's dead, right?
Will you zoom in on that? Will you zoom in on that pretty big, just so that people can kind of get a general glance of what we're talking about here in terms of the.
This is the complexity of a eukaryotic chromatin, and I'll show where the histone is on this. It is a little blurry because we can't fit everything. Even the author who put this together obviously had some problem. So do you see this dark line here? These two lines that on the left here. And as I go, that's the DNA and it's winding around. These are the histones here. This is all the machinery that is interacting with the histones to make the histone work. If there's a double-stranded break in the chromatin, it has to do all this remodeling and signaling and moving histones aside and then marking them and flipping bits on and off. Just massive amount of computation to do a repair. If you think about this, if you took an axe to your computer, wouldn't it be cool if it could repair itself? This is what it's doing.
Well, and that's one of the things that I find extraordinarily fascinating listening to the atheists on their diatribes. They constantly try to use copying errors, as somehow a argument against the designer. Oh, if they were a designer, why does this happen? And I'm like, okay, number one, you guys are not calculating in the repair process that occurs when you're looking purely at like in transcription, the error rate, or in ribosome. I think ribosomes are like 1 in 1,000 wrong amino acid comes in. But yeah, then it's fixed though. So then when you account for the correction process, it's actually equal to, in many aspects, better than our own copying process in terms of error prevention and data corruption. And then that's before you, and this is the point I really want to make here, is you talked about taking a hammer or an axe to your computer and opening it up, right? What I think so many people do not even comprehend or consider is that if you removed all of the shielding from around your hard drive and attempted to copy or even just let it sit there with that shield, the data corruption would be massive in a very rapid, very short period of time. And given your background on this, would you agree with what I just said there?
Yes, in general. And I want to point out there are a couple things. When I said you take an axe to a computer, when Dr. Sanford developed his gene gun, people thought he was, it's funny to hear the anecdotes because he was there in Cornell, and the first gene guns actually used gunpowder. So there at night, you'd be hearing the sound of guns going off.
It wasn't figurative. It was literal, huh?
Yeah, the first, that was way too powerful. I mean, they're blowing apart the cell. And I would suspect the reason people didn't think it would work is they're basically taking DNA and shooting it into the cell. It's like, well, why would you expect that to work? So they put it on gold, and then they found a way to basically shoot it. It's like a BB gun with DNA in it. And why would you expect that to work? Well, superficially, we wouldn't think it should work. And I said, John, how did you not blow apart the cell? He said, actually, we blew a lot of cells apart, but some of them would survive. And the way that they're able to survive is you have this free-floating DNA that was just shot in, but the DNA repair mechanisms could incorporate it. We probably still don't know exactly how it did it, but without that self-healing mechanism, the gene gun would have failed. So, sorry. So the moral of the story is that I was exaggerating, obviously, about taking an axe to a computer, but on some level, there's self-healing. The second thing I wanted to point out, and when the atheist says, why do you put errors? Well, maybe some of those atheists are not electrical engineers that do hardware design. Mm-hmm. The compact disc.
It's a fundamental you guys always account for, right?
Well, the compact disc, this is why Claude Shannon was famous. So we talk about Shannon information. People credit him with the bit, but that's not what really made him famous. What made him famous was that he realized that the most compact way to store things is to store it, have it with errors, and then correct it on the fly. Because if you think about it, if it just pencil and paper example, if you write real small, you're liable to make errors, but you can press more things. So the question is, do you want to write real big and have no errors, or write real small, have errors, and then correct them on the fly? Shannon's famous theorem on communication solved that problem. And he established the limits of the amount of errors that you tolerate. Really, the correct term is noise, right? And versus the signal. But the best way to architect a data storage machine is to create errors in it. You have to allow it. That's why your compact disks have what's called Reed-Solomon coding, because they realized, no, it's going to be wasteful if we make a disk that'll never make errors. You could hypothetically just have a compact disk that had one bit. It's never going to make any error. But
I'm assuming if you try to go down a 0 errors allowed, you're probably dramatically slowing down the replication process too. Would you not?
Yes. So that's my counter to the atheists. I said, you guys just are not very sophisticated, because it depends on what you're trying to optimize. If you're trying to optimize for compactness, you want errors. In terms of, as far as if you're talking about this universe, that's the way things are going to work. So, you might hypothesize an alternate universe, but then, we're getting into really strange philosophical territory. But the question is, we really shouldn't be trying to use that. They shouldn't be using that as a bad design argument. That actually echoes 21st century knowledge of information theory and data storage. There's error correction mechanism. That's a genius way of putting it, because that's what our best engineers will do. That's what happens on the internet. That's what happens with your modems. There's error correction at all levels. See, they could say the same thing. It's like, well, why don't we design modems that don't transmit any errors? We don't do that.
And to kind of put this into context in terms of, I mean, obviously everything you're talking about here applies to a plethora of aspects of biology, but just in the context of what we're talking about tonight or today of, gene expression, I think people often forget that in the polymerase, there's literally an autocorrect function built in. And I mean, people forget this, that as it's flying across through, flying down a sequence, it's being able to identify and autocorrect. And the same principle applies to, from a different context of, mutations damage that's happening to our DNA on a daily. I mean, millions of times a day, something goes wrong due to radiation or whatever it might be, and we have physical protein machines that go and fix where the damage is. And to me, when you think about that, it takes things to a whole nother level when you contemplate the premise that, how do these things know that there's an error? If this is nothing but, chemical reactions, how are these proteins being able to identify that this is now no longer correct? B, go grab from a separate location, go get nucleotides to bring back and then or put in the correct order and reinsert into where the damaged DNA was. And to me, when you contemplate, and correct me if I'm wrong on this, but I'm pretty sure it's a well-established, accepted premise that if those functions did not exist, we would die so rapidly from gene collapse.
And I'll tell you why is. When there is base pair damage, this is like cutting the wire. It really is like cutting the wire. It's done, or like when you break up a railroad track, the train can't move any farther. It only takes, we might tolerate a certain kind of point mutation, but the DNA damage that happens naturally is the stuff that'll break the readability of the helix. Because just imagine that you have a piece of tape and it has all this lettering on it. Well, just imagine taking a piece of scissors and cutting it.
Right.
You're not going to read the rest of the tape. That's the sort of damage that we're dealing with. So I just sort of laugh at these people and think that they're going to synthesize these nucleotides randomly, and you're going to have a genome. It's just like, well, it's not going to be there for long.
A quick, as you're talking about the tape and cutting it, something that sparked another thought in my head. So something that I have been dealing with recently is and I think this is heavily due to modern tech being all digital in the modern computer technologies perspective. But people seem to forget that and just dismiss the premise of the information transfer that's occurring in the spelling of these proteins and all the other things that we're talking about today, because it's a chemical medium. And I think they don't contemplate or even consider the premise that, hey, we used to use physical punch cards, magnetic tape, and other things to execute or to both store and then be expressed, for lack of a better word, for the end function, right? And I think so many people, especially people 30 and under, for example, because they've never even, that's such a abstract thought. They don't even consider the comparison that we're talking about here not being just purely analogous, but literal in terms of the expression of the outcomes via what we're talking about now.
Well, I know that you like the information argument, which is what I call kind of the software side, and I argue the hardware side. Right. The way that I argue it is like, okay, let's just say one is a pure materialist and doesn't think information is anything more than kind of an abstraction in our head. And we know, you and I know certain people like that. Just first off, it's understandable why ID proponents would kind of go toward an idea of something that's transcendent of material reality because it echoes. But let's we don't even have to win that argument if there's kind of a duality to reality, you know, hardware and software or hardware and information. We can just start and say, let's say it is all chemical reactions. Let's just go by that assumption. There can be lots of chemical reactions that don't work, that don't make cells. We can imagine a bazillion ways that there's not going to be any replication. Just try to say, let's make it simple. Think how hard it would be to make a chemical 3D printer. A chemical 3D printer. That's the basic problem. We can barely even do that in the macro level. These, you know, some of these factories finally started creating 3D printers, but it is so much harder to do it at the molecular level. And what we're doing duplication of what Dr. Change Tan said, who wrote that book, Stairway to Life. We were talking one day and she said, everyone thinks, you know, it's just all DNA. That's the easiest part to replicate. You can't replicate the cytoplasm that way. We can't do that in the lab. We don't know how.
And then you have to get into the lipid bilayers and all those additional pieces that are. And I agree with you on the. I totally follow where you're going with this in the context of. Yeah, well, there's like exponentially greater, more complicated things than just the DNA.
So let's just say, okay, I'm not going to even argue information. Tell me if you could build something like this.
Right.
Can you build a 3D printer? Again, this is like a Rube Goldberg machine. It's like, I'm not saying, you know, like these people build these RNA replicators. I say just, okay, that's all well and fine. All right. So anyone can build a paperweight. So I call that a paperweight. It doesn't take a lot but build a 747. Build a machine like a bird. Oh my goodness. This is like an airplane, a miniature airplane. Some of these birds are miniature airplanes that can replicate themselves. Build that from scratch. You tell me, you just, even under your materialist assumptions, you try to build that and you'll see just how daunting this problem is. It's not so easy. So you could just say it is chemicals, but you better pick the right chemicals. You better put the molecules in the right spots. Then it's a different story. Otherwise, you just have something dead, and we know that.
Were you? I'm sure you picked one, but will you pull up one of those slides on the DNA folding things we were talking about?
I'll show you that.
Because I think what you just explained there is visualized by some of those different components with the transcription factories and everything.
Right. I'll show that. Do you know what this one is? This is one of my favorite slides. And speed of sound used in the comment section. This is man-made memory here. He said this looks like the kind he had in his computer in the 1970s. And this is an abacus from ancient ways we did memory storage. This one's the God-made compute. This is God-made memory. I just couldn't help but notice the analogy. That's really cute.
Yeah, I heard a very interesting comparison of all this stuff.
Beads on a string thing here. You see that? You see the theme. So these are the histone nucleosome things that we were talking about earlier, where I depicted it like this. But the way it looks like on an electron micrograph is something like this. It's the same thing. Right. And so you see these beads here. You can barely make out the wire. That wire that's connecting them is the DNA. So you requested the 4D nucleome. Let me see if I have pictures of 4D nucleome. Here we go. So if we have a robotic assembly line, you have all these little arms here, and it has like the tools or machine. You know, there's a machine and a tool to help put things together. Well, in DNA, it should look very similar. So this here is DNA right here, and it can be folded, and then proteins can attach to it that act like tools. For this to work right, this is where there has to be foresight. The spelling of the DNA in this section has to be something that the correct machine will recognize. Otherwise, you're going to get people parking in spaces that they should be parking in. So the spelling of this section of DNA has to match what the protein will recognize. This will not happen by random accident any more than a lock and key. Random lock and keys will be matching. It has to be deliberate. That's my inference anyway.
And quick interjection on this.
Right.
Can you move your mouse over the coding part of the gene? Just kind of okay. So for everybody looking at this,
this is the coding part of that's
what the atheists, evolutionists, etc. say is the only real component of life and their evolutionary models. And earlier I made the brief allusion to the fact that many of the other things that enable that minuscule source code component to be in comparison to be actually executed and expressed is all these other factors that are way far away. And now, as Sal is showing us here, not only are they linearly far away, they have to be brought to a place physically in both 3 dimensions and the fourth of the time factor because if they're not, then what happens? Nothing, right?
Well, you get really bad things that'll happen. Like, for example, the lenses in your eye have certain kinds. It's a certain cell type. It's different than your bone cells, and your bone cells are different than nerve cells. The regulation is really important because it basically every cell in the human body has almost an identical genome, but it has to express it different ways so you can get different cells. It's not exactly identical, but I mean, that's a good approximation. There's some changes that happen in the somatic cell line that we don't have to get into today. But so these machines would be different depending on the cell type. And there has to be a lot of if you think about it, think about all the computation that has to be done just so the cell can figure out, oh, I'm a cell in the eye. I have to behave like a lens cell. And that's interesting in and of itself because the lens cell eventually dumps its nucleus so that it's even clearer. I mean, they're just whacked out things that happen. And in the heart cells, it's polyploid. There are a lot of cardiac polyploid cells. So when Darwinists say, well, what's the good use of polyploidy? Just like no heart researcher would ever say that polyploidy is junk DNA because they're actively researching it. They know it's there for a reason. They suspect it.
Quick interjection here. The point you were making on the getting rid of its nucleus. I think flies over the average person's head of, okay, folks, let's think about this for a second. This machine component builds itself. It has all of these different facets and components to it. Then it ejects the CPU.
Yep.
And the hard drive and everything ejects. And then it still works.
And then even its organelles
continue to function.
But the way it works is because that's why you can see, because it's maximizing the ability of light to flow through it. And I'm just like, yeah, I think about that. It's like, yeah, Darwin, he said, these are organs of extreme complication and perfection. He said, when he considered these things, he said, my theory looks absurd. And I said, his first intuition was correct. It is absurd. I mean, he managed to talk himself out of what he had, right? But so this is what they call cis regulation, where this is happening on the same gene, same chromosome. There is kind of cis regulation on the same chromosome, but different genes. So you'll have one gene, like say machines on the introns of one gene regulating the exons on another. I mean, I've seen diagrams of this. This is just whacked out. I mean, just the ingenuity.
So you're saying that a supposedly non-important component, such as, or what's called non-important, i.e. introns, up until relatively recently, right? And even by some still dismissed. You're saying that those are the anchor points, if you will, for these machines that enable the expression of a specific gene, but those might be on the supposedly non-important introns in a completely separate gene sequence. Is that correct?
That's correct. The intron of one gene could be controlling the regulation of the exons on another gene, and it could be millions of base pairs. This is what they call enhancers. And when I started to see that, I said, no wonder the evolutionary biologists were so angry, because it looks ingenious. It looks ingenious. We begin to look at the role of DNA is no longer, like you said, it's not necessarily the atheist. Probably the more correct term is the dinosaur neo-Darwinist would only want to frame it in terms of this region here.
Right.
The modern ones like Kunin, they know this is serious. This is seriously difficult to explain. They know it. But maybe once in a while they'll speak up and say this is just way too complex. I actually, it's really funny. He works for the National Institutes of Health, so he's been curiously silent on the whole ENCODE debate because he heads up their evolutionary genomics thing. And while Dan Graur was just trash talking the NIH, Kunin was strangely silent. It's like he didn't want to offend the evolutionary biologist, but yet he was on the payroll of the NIH at the same time, and he probably knew the NIH was probably correct. So right. That was a very interesting battle.
Oh, I can only imagine, especially given the fact that so many of these people have been, at different stages in their careers, have made extraordinarily adamant arguments about XYZ has zero relevance. And then fast forward not too long later and they're having to concede. And now you get to the point where they've been in the field for 30 or 40 years. You have to imagine that the gut punches they've taken over the years in terms of new discoveries have got to start. I mean, they got to be in the 15th round at this point on denial of the obvious. Oh
yeah, I mean, it must hurt that they're kind of the laughing stock privately. I mean, not publicly. They're always, the media and is committed to a certain narrative to always lionize the evolutionary biologists. But privately, people are like, "You guys are worthless." I mean, that's a mean thing to say, but I'm just like, I could look at some of the top research groups out there, and I looked at their resumes. I said, the one that's constantly missing is evolutionary biologists. They have chemists, they have physicists, they have computer scientists, engineers. We're the evolutionary biologists, they have. It's just kind of embarrassing. But if you think about it, what value can they add? One of their best evolutionary biologists is saying this is all junk. What value will he have for cancer research when they're looking at this stuff that he's saying junk and he's saying you should disregard it? And the cancer researchers are saying, no, well, we have a problem with that because that part of the DNA region is associated with cancer. We need to find it out. We don't care what your ideology says. We have a job to do. We have lives to save. So, buzz off. So that's basically, that's the contribution of one of the best evolutionary biologists out there. And that's just totally pathetic.
Well, in line with what you're saying there, what I find so interesting about, one of the things I find very interesting about what you just said, not because I agree with you completely on the evolutionary biologist, I find it very interesting, different ones I've interacted with offline and, face-to-face conversations. Some of them, it's like, if I know more about some of this stuff than you, and you have a PhD and you don't understand some of the basic fundamentals, that's a problem. But where I'm going with all that really is, you talked about engineers, chemists, physicists, and computer scientists all working together to try and figure all this stuff out, right? And to me, what that showcases is that we have facets of computation, mechanical engineering, and then understanding of physics and chemistry being required to have the possibility of understanding it. So if that is true, then it requires all of those components on the molecular level being existing for life to exist, and to argue that you have to be an expert in all of those different fields working in unison to have a chance of being able to actually truly figure out how this all works, to argue that it is not reasonable to suggest that a supreme intelligence was required for this, to me, as I love to say, requires suspension of rational thought.
I try not to. I try to give people the benefit of the doubt, and all I could say is that's just too much for me to think this is an accident. It's too much for me to think this could be done by natural selection. It's too much for me to think it's anything except God.
Yeah.
And
wait, in line with that,
it would take more faith for me. It would take a bigger step of faith and self-delusion to say, I think this can happen by ordinary mechanisms. Something very, very special put all this together. Oh, by the way, here's a picture of one where you had genes co-regulating each other. This is just one of them. But I have better pictures elsewhere. But this is showing a little bit of how they can associate. They're actually better pictures than this. I'll have to gin them up. But then the one that's really whacked out is when they're across other chromosomes.
Yeah, I remember the first time you showed me this concept, and it was not only because I had some basic information or basic knowledge of the DNA folding and stuff. I had no idea about the interchromosomal.
Yeah, transchromosomal. Oh yeah, that was a big deal. People didn't think that would happen.
That's a mind bender, if you ask me. I mean, if you don't think that, what? I mean, to me, that also completely damages, if not destroys, this whole, there's no significance in the position of genes inside of a chromosome and how they compare to another organism.
Exactly.
I'm like, no. It actually does matter where it is in the chromosome.
Right. And let me tell you why the sequences matter. This is the fire link RNA, which they thought link RNAs were junk, by the way.
And that's the long non-coding.
Right. Long intergenic non-coding. Some spell it LINC and some just spell it LNC. There's probably a subtle difference where I think LINC is a subset of LNC, but we don't need to split hairs on this one. But if you think about it, how does that link RNA pictured in red here bring the right region of chromosome 17, the right one of chromosome 15, and the right region of chromosome 2? It has to be sensitive to the sequence here. So sequence matters, even if these are supposedly junk DNA. It's doing something to recruit it and position it. The other thing that I didn't mention in some of my other talks, there are all these structural proteins that help the fold. They call them structural proteins. So those have to have the right spelling. Nothing attaches to anywhere unless the sequence is correct. If you get the wrong sequence, the machine is just like going to the wrong parking space. If someone, like in some of these parking lots, they have numbered spaces. If someone went there and maliciously started renumbering it, that would be bad.
So to make sure I am understanding this correctly, the link that you're referring to must,
this link RNA here,
the link RNA must be appropriate to attract not one, not two, but 3 different sections in completely separate chromosomes.
Right.
Okay, so you can't just, oh, cool, it does this one, and then the number from where I'm addressing is the cascade argument of, well, if it connects with 2, then 2 connects to 15, then 15 connects to 17. No, link RNA in chromosome X must connect with all 3 at the same time.
And actually, the diagram is a little misleading. You have actually a concentration of these link RNAs. Again, we don't quite know how it concentrates, but it seems to be associated with it, drawing them together. So you could see that this is a big geometry problem. Right. We can't just be inserting DNA randomly and expecting this to work well. And that's why it's translocations in the chromosome can be really damaging for higher higher eukaryotes like us. There might be animals that might be able to tolerate it, but for one thing I wanted to say is there seems to be a stairway of complexity where the lower creatures can tolerate certain kinds of changes. Humans, we're kind of at the limit as far as it's going to affect us radically. We're sensitive to changes like that. But one thing I wanted to mention is this conformation here will change depending on the cell type. That's how regulation's done by geometry. So it's not just this. Just think of all the, they say there's like 300 or so canonical cell types. That's just a classification convenience. There could be really thousands of different. And it wouldn't surprise me, like in the brain, every brain cell is different. But if you think about it, how do we remember anything if you don't have unique addressing? So every brain cell has to differentiate itself, and then it has to sort itself out so memories can be remembered. Not just like emotional memories or recollections, but just think about when you taste something, when you smell something, it's like, yeah, that's familiar. I know what that is. So every experience we have is recorded somehow. That's not a trivial storage and retrieval problem.
Well, it's interesting when you're talking about that in the context of just the data storage in and of itself. I find it very interesting how, and this goes back to the dismissal, not only of the all of this amazing engineering that we've been talking about for the last 30 minutes. Then when you factor in just the information storage component from the hardware side of, this even better than I do, but pretty much every major tech company on this planet is investing significant capital into trying to figure out how they can not storing the data is easy. Getting it back is hard inside DNA. And they were trying to create all these machines to basically be able to do what the transcription complex that we've been talking about today executes. But the most recent projection I saw was that they could store every byte of data on the entire planet in DNA the size of a teaspoon. And that's how much data can be stored inside of this thing. When you think about it from a, wow, holy crap, this is an amazing storage medium. But then when you factor in, and as you're talking about the individual cells in our brains, the most recent study I saw on it was they project that our brains individually can store about 2.5 petabytes of information. And I would wager that's probably a underestimate because that one was 10x greater than their previous assumption. But even if you just think about 2.5 petabytes of data can be stored in an individual brain, the power consumption requirement, which is negligible compared to storing in a human-made hard drive a fraction of that.
Yeah.
And I think people just don't think and consider these facets of, and how there's a point you've been making of the unison in which they must work in a physically structured environment.
Well, you highlight something there most people don't comprehend, which is sad. That's I feel one of my missions is to make this a little bit more accessible. I don't want them to have to go to graduate school to learn this. They don't have to. See, you and I are talking about this, and I could expect that if I just had a few days with someone, they'll start to see this. They don't need to spend, not that there's anything wrong with spending 4 years in a traditional university if you're going to be an organic chemist or a biochemist. You would need to suffer through 4 years. But to appreciate the designs and the wonder and the beauty and the general wow, this could be learned in a week. That would change. There are probably people out there that this would make the difference in their life as far as believing God. There's one verse that maybe I kind of dismissed when I was very much younger that's meant more to me now. When Jesus was saying, don't be anxious, how did he open that? He said, consider the lilies of the valley. They neither toil nor do they spin. But I tell you, Not even Solomon, arrayed in all his glory, was as one of these. And I think for thousands of years, maybe people just said, "Well, that's kind of a cute saying." But here in the 21st century, in light of what we know about photosynthesis and how cells work, that is exactly true. We cannot if we took all the riches in the world, we could not make a lily of the field with all its mechanical complexity and photosynthesis. We can't build it from scratch. And I think that verse has more meaning. And I believe Jesus Christ is the creator and he put this together. So when you say the average person doesn't see this, that breaks my heart, because they should.
Well,
because now, yeah.
Well, no, I agree with you completely. And just that slide you have brought up right there on all of and that's obviously just a fraction, as you mentioned earlier. There's no less than 300 other types of cells just in the human body. All coming from stem cells. And then the fact that people discount that, and we'll have to have another stream about this, because we couldn't cover this in remote detail without talking for 3 hours. The fact that not all, but a huge component of how those different cell types come from the stem cell is from epigenetic markers. And then to me, when you contemplate that in the context of, is evolution logical, plausible, and probable, if your foundational argument is undirected mutation over time, but then the thing that we now know accounts for something like cell types is directly, in no way, shape, form, or fashion caused by something that happened on the purely DNA sequence perspective in the coding regions. I mean, it's almost like a borderline joke at this point when you look at that. And correct me if you think I'm wrong on this, but to me, that in and of itself destroys maybe not all, but a massive element to the evolutionary argument. And then while simultaneously inserting gigantic evidence for the foreknowledge requirement for life to exist.
Yes. And there's kind of an unfortunate conflation, what they call common descent versus the design. I don't mind. I mean, that I'm a young earth creationist, but I don't mind saying that. Let's just say, for the sake of argument, let's be like Michael Behe and just assume common descent. You still can't explain. Common descent does not explain the complexity. Common descent is just an assumption, but then for common descent to work, it's like, well, you'd need miracles along the way to make it work. Because
in line with that, something that they oftentimes try to avoid is the variation. Through morphology, they try to avoid the body plan differences and the requirements. And for so long, all you heard was, Hox gene.
Oh, goodness.
Well, I was reading a paper 2 days ago on the stunning discovery that, holy crap, all of this junk DNA that we said didn't have any bearing, oh my goodness, it seems to have A, variations between different types of organisms, but B, directly influence the outcomes of the Hox genes. Therefore, it is not just the Hox gene by itself that is responsible for physical structure of the, insert whatever organism type. And as I was reading through that paper, I was just cracking up. Really? This wasn't obvious before? I guess you're now coming to this conclusion. It's like, let's think about this, guys. You've been arguing that the exact same gene with the exact same data in it somehow results in dramatically different organisms. In what other context would you argue that is the logical conclusion? There is none. There is none. You would assume there must be an additional variable we have not yet considered. And yet, for probably no less than 40 years, that was argued as the fundamental, one of the fundamental defenses of neo-Darwinism. And now they're having to concede that it is actually works against them.
They're getting crushed. I wouldn't want to, that's why, they have this third way of evolution. Just out of curiosity, do we ever take questions from the audience? Thank all the people that are here.
Yeah, if anybody wants to ask questions, shoot them our way. That's I know a couple people dropped off. We still got a few.
Yeah, we probably nerded them out. So we need to we need to be able to have like kind of a. What I was thinking is maybe creating a set of lessons where they're taught top down, where they can just go real high level, and they don't need any prior biology, and then they can kind of hyperlink to learn little smaller pieces along the way. Because to be throwing stuff like this, even Dr. Sanford said you're going to lose even some of the PhDs because the field is moving so fast. The way they learned it 20 years ago is like obsolete. And then I talked to a professor recently. He said, "Yeah, Sal, even 10 or 5 years ago, what was textbook is it's getting obsolete really fast because they're throwing so much money at all the research trying to find cures for diseases." So this is kind of the
and that's why I think that's part of the reason I wanted us to kind of visualize some of the things that we've talked about of the 3 and 4 dimensional aspects of gene expression, not just being it's just a chemical reaction. Like, yes, there are chemical reactions that result from these previous actions, and you briefly mentioned them. I think you called them is it structural proteins that actually are doing the bringing things together. I mean, to me, when you think about that, it's folks, you have a mechanism that is not yet chemically connected to the other one that brings it to connect with the other one. And you're claiming that this is just happening. It just happens. And there's not a predetermined elements that are required in the original spelling that we started this thing off with that result in these components that can ultimately fit together to result in an end outcome that results in another mechanism like the one or similar to or completely different than the one that was used to enable that process. And I think people don't really consider, I talk about like in a context of chess. If you're going to argue pure chemical reactions, then you barely know how to move a pawn one square forward. You sure as heck don't know en passant, and getting into any kind of, learning openings in encounters and all that kind of stuff in chess, you don't have a clue. But you're comparing what's going on in biology to, oh, cool, we moved one pawn forward, and then thinking that explains the chess master who can think 17, 20 moves ahead. Anyway, enough of my little diatribe there.
I'll show you some of the structural proteins. I believe these are cohesin complexes here. They're like little rings. That's kind of interesting in itself. How does a ring get on the DNA? That's a whole nother story. But that's some of the structural proteins, and there are many more that help build these loops that create all this regulation.
And don't those have to be facing, like, there's 2 different types, right? They go different directions.
Yeah.
And
let me see if I have that in my slide.
They have to be facing the right direction.
Oh yeah, let me see.
For the DNA to be going through, otherwise it doesn't work.
Yep, here it is. Here's the slide. Let me see if I can get rid of this. Okay, so I just kind of showed. I showed this diagram here. You see these loops? We can call them an extrusion loop here. So now I'm just going to put this in the PowerPoint, and you can see kind of my rendering of that loop. So the DNA. So this is all DNA. This black here, this is DNA. And I don't know that the gene is actually there. That's something I have to talk to people about that. So this may be a little mistake here. So I might just delete that from the slide anyway. Because I actually, let's just say I don't know exactly where the gene is, but just let's say this is the regulatory loop right here. So let's just call this an arbitrary read direction along the green. So it's going like that. So when we're reading on one strand, it'll be, you have to have the sequence. It's CCA, CNA, GGT, GGC, AG. That's the consensus sequence. But when you come up over here, it has to be the reverse complement. And that's what I mean. It has to be matched. So you could say that the DNA points this way here and it points this way there. So you could see the red arrow is in the same direction as the green arrow, whereas here they're in opposite directions. And let me go to the next slide. I can show you a diagram of this. Maybe I'll play it's only a minute video. You could see what they call loop extrusion. So the point is for this loop to form, one needs to have this little consensus sequence here in the right spot, and then the consensus, the reverse complement in the other spot, which is basically kind of like the reverse of it there. Why does that pair? And we have so many pairs like that. I mean, it's just like since you program, you know the importance, like in certain languages, you want to match braces or quote marks together, but especially braces. You have a left brace and a right brace. Mm-hmm. In the C language, you better have them all matched up. Otherwise, you have a disaster. The genome's like that.
Right. Wait.
And this is the average length, by the way. This is 200,000 bases, 200 kilobases. You want to have this, and then 200,000. You want to have 200,000 bases later. You want to have that sequence. And this isn't just in one location. There's so many of these. And where are they parked? Many of these are found in sine transposons.
Explain those transposons to people that the average person isn't going to know what that is.
That's another thing that they call junk DNA, that they called parasitic DNA, that just made a bazillion copies of itself and filled the genome. People thought that was junk. But now we're finding out some of its function, and one of its functions in many cases is to create these loops that do all this fancy regulation that basically create machines like that. You know, kind of in the man-made world, we have in the God-made world, that's how these loops are formed.
Right.
So this isn't even showing, you know, you could just imagine it needs to have those letters in it. So maybe you could talk a little bit while I look for the chromatin extrusion model.
Well, you know, what you're talking about there really, it's intriguing as well as entertaining when you think about the desperation with which things like transposons are leveraged by I'll name somebody who I've gone up against, R.J. Downard, that's one of his favorite things.
Sorry to laugh.
As supposed proof beyond all doubt of both evolution as well as theistic models of intelligent design. And I'm like, okay, well, number one, we're discovering things about them like you just described. We're also discovering that some of them are literally, especially during embryo development, are literally taken from position A, moved to position B, reinserted, modify the outcome of sequence or sequences, and then when that temporal factor is removed from the equation, then that same thing is moved to a different location. And to me, when you think about that, it's a mind bender. I've actually seen this one before. This you're talking about on the extrusions, right? Those rings going in having to face the opposite direction.
I think this is the cohesin here. Oh wait, let me stop it. It just pointed to it. Remember, I gave that consensus motif.
Mm-hmm.
It says CTCF motif. That motif looks like this. The CCAC. It's not exactly this, depending on which author. It'll change by one letter because it is a consensus. So when I pointed to that, just imagine this string here is this region right there. And so what they're trying to point out, see, it's pointing one way, but then you need to have it 200,000 base pairs later, have one that's paired to it pointing the other way. And it shows it won't recognize if it's pointing the wrong way. The protein is smart enough to know, oh, I'm going to ignore that one for now. But depending on the cell type, it can pair them up differently. And we don't quite know how it does that. But that's really cool because then you can get different loops. So this is like a C program with the braces, but you can dynamically change what matches to which is just totally mind-blowing. And the reason I like this video is one can see, yeah, it doesn't look too it looks complex enough when you just have one loop. But when you see it in the context of the entire, like, say, nucleus, it's amazing that all this could be coordinated. See, yeah, one begins to see this, what we can call colloquially Brownian motion. All this is going on, and it looks like a chaotic mess, but it's very organized.
Well, what's interesting, I think they actually talked about in this video, if they were not going in the appropriate directions, then you would end up with everything tied in knots, basically. When they are going in the appropriate directions, then the loops fold out perfectly to enable, or perfectly is relative, obviously, but fold out in the manner in which the genes that are housed therein in that section that's being unwound are now being able to be expressed. And
yeah,
I think they're showing that right there what I'm talking about. Of, oh, hey, cool, it works
can detangle,
yep. Which
is just we can't. You know, when I try to tell guys, I said, "Do you think you could build a machine this complicated?" And the organization changes between cell type. That's how gene regulation works. I mean, you could just how hard we had to work just to be able to even begin to understand this. This is why we needed computer scientists to do this, because we had all this data and we couldn't map it in 3 dimensions. We had lots of little pieces, and then they had to it's like a jigsaw puzzle. They had to throw it in supercomputers, and then they could build a 3D model like this. So sorry, I'm just dissing evolutionary biologists, but what Dan Grauer did just left a really bad taste in my mouth. You know, why would he be calling all these people that do work like this ignoramuses and crooks? That's just evil. So it left a really bad taste in my mouth. I said, if he's doing that to medical researchers, how much more is he doing it to anyone else? You know? So.
Yeah, it really is as you're showing these different things, we're discussing them. I mean, it's unavoidable that there is foreknowledge requirements. It's not just, over time, this all could come into being.
I love, like, don't you love that picture? Look at the complexity.
And it has to be able to be expressed on the fly.
Right. Their genes here, their polymerases just zipping about, transcription. They're not even picturing all the other machinery that's here. I mean, you think about the spliceosomes and whatever else is, they're floating around in here too.
Let's see. Something, I want to see if this will come up. Hang on. Let me share my screen real quick.
All right. I'll stop my share. So thanks to the audience for forbearing, since my screen was shared for most of the
all right.
Most of the talk.
Let me know if audio comes through on this one. Let's see.
So in the meantime, Redefined Living says, Matt, do you do any clothing design for SFT? Do you use one of those YouTube merch stores or pre-purchase? And sorry, I just had to mention that.
I think he was mentioning wanting an SFT, a hat with the SFT logo on it. Let's see. And I'll let SFT and Matt know if they're not still in the chat. They need to get you one of those. Okay.
And he said, because I just want a gray hat with the SFT guy logo. I'll help pay for setup or whatever. Email me if you guys are interested. And Little Yak says, you.
Let's see. All right, people, is this coming through?
Yep. No, I just wanted to mention, I can't seem to expand this to fill out my screen. That's okay.
Let me know if that's coming through. What you're seeing there is the beginning of the different transcription factors and complex that are getting ready to. They're all coming together to get ready to start to transcribe a gene. And obviously, they're not even. If you're, I don't know if the audio is coming through, but in the audio of this, they're actually talking about how they're not even coming close to showing everything that's going on. And then here comes a fold. Boom! Everything is turned on, and now the polymerase is zipping across, transcribing. And something they talk about is this is about as close to real time as this is not figurative. This is a literal representation, both from a what's happening as well as a time perspective. So that yellow thing you're seeing shooting off there is an mRNA. And all the other yellow things you see flying in, those are nucleotide bases. To me, when you really think about this, and earlier we talked about the autocorrect. There's actually, they don't show this in this animation, but there's actually an autocorrect function that's going on. If they make errors, it like on the fly fixes them and stops and stuff. It's nuts. What is able to be done. And then when this process is, this is like not barely even step one of what happens before you get to the different things to make it a mature mRNA are executed, and before it even heads over to the ribosome. I mean, there's so many different modifications.
Oh yeah,
that are done,
you can keep showing it. By the way, that's fun to watch. Like you said, whenever you have doubts about a designer existing, you just look at that.
And then when you look at, I'm going to show this other one. And again, what I'm about to show you is dramatically dumbed down in terms of the actual complexity that's occurring. And so now we're going to talk about getting into, okay, so it's left, that mRNA has now left the nucleus, and it's heading over to, where is this going? Okay. Now it's coming through the cytoplasm over to, so it's left the nucleus. And by the way, those pores it came out of, there's like a whole aspect of what's happening for it to even get through there.
Transmembrane proteins.
But here, I'm going to put this on pause real quick because this is something that just irritates the fire out of me by the atheists. When they talk about there is nothing about the translation process that is formal in regards to how a codon equals amino acid and then is somehow brought together by multiple other components. So you got the mRNA is processing through one codon at a time through the ribosome. There, the red you're seeing is a simplistic version of a protein being folded.
Polypeptide, yep.
But here's the process. So it binds. And again, this is dumbed down. There's way more than this going on here, by the way, folks. There you've got the mRNA going through into a machine that is recognizing it. Then we have tRNAs that are coming here. On the red little dots are amino acids coming in. But on the bottom of on the other end, completely separate from the amino acid, is the anticodon, which somehow those came into existence when you think about it. But they go and match up with the codon on the mRNA, and then and then the ribosome also has processes by which it checks to make sure that even if the anticodon match is correct, it actually has the correct amino acid, which is equal to the one that's represented in the codon. And we're really dumbing this down, but ultimately one of the parts that just blows my mind is that, by the way, you're only seeing half of this. There's actually another component that completely covers this up. And there's an entry point that is perfectly angled for the tRNAs to come in. I mean, it's like a completely perfectly shaped doorway for this machinery to come in. and you're talking about A site, P site, E site. But a fundamental piece, I touched on this earlier, is that amino acid that is being part of the protein synthesis process never chemically interacts with the codon in the mRNA that is coming through. It does not chemically interact. The codon does not become the amino acid. They are separate. And
one way to make your argument, they've reprogrammed the genetic code in some of their synthetic biology.
Yep.
So there's no inherent chemistry that requires it to be one way or the other.
Right. They've created six base syntaxes. I mean, they've done all sorts of different stuff to store pictures or to create new outcomes.
I didn't know that, but I guess what I was just trying to suggest, that's a different way of saying it's not chemically interacting.
I agree with you 100%. I'm like that's the point I'm making too. And I agree with you. It's a great point. When we've been able to create our own syntax for completely different outcomes, whether it be pure data storage or to try and modify, create our own custom little bacteria and stuff.
Well,
it's unavoidable. It's just it's a fact.
What a researcher told me is that when George Church modified the genetic code, and their safety reasons, they're trying to do this because they're actually concerned about genetic engineering monsters that can escape out of the lab. They wanted to have some way of like self-terminating. I don't quite know what it meant. I think what they were trying to do is they said they're going to give it a different code because in case the thing escaped, it wouldn't be able to prosper in a natural environment. I don't know all the mechanics of it. I saw the lecture. It was phenomenal. But what one researcher told me, he said, well, that code doesn't work so well. The thing is pathetic in the way it's able to do transcription and translation. I don't know all the details, but that was...
Well, it's interesting you're talking about that on the not being able to work. I actually read a paper on... This is one of the things that cracks me up when people try to argue that there is no... And I know from your perspective that I think the words you used were the information arguments are almost sophomoric. So many of the ones that try to avoid the premise of there even being one, right? Or there is any information at all in there. That just cracks me up. And I was reading this paper done by some atheist origin of life researchers, and they were comparing. They did a whole study on whether or not the quaternary code base of the genetic code was efficient. And was it the best for data storage and stability and robustness and all those different factors, right? So they did this huge study to come to the conclusion that while there were a few other syntaxes that might be better at this piece or that piece, what we all agree enables life to exist is the optimal. And from a both a mathematical and obviously just technological perspective and an information theory perspective, all those different factors, it was the most efficient. And it was pretty entertaining in the conclusion. You could almost feel their frustration with being forced to report this being the outcome of their research.
Yeah, I mean It never ceases to amaze me the lengths people go to argue that life is junk, that their life is junk. I mean, is that your purpose in life, to show that you're poorly made? And I just like, granted we're frail, we're fragile, we're subject to disease, but there are aspects of when life is at its peak, like our eyes get weaker and all, but when it's at its optimal, it's an exceptional machine, and it does make, in the evolutionary biologists, it raised the question, why do we even die in the first place? And it's because, and I know why they think that, they're creatures that don't die by natural aging. The Hydra is a eukaryotic multicellular creature, and I only learned this like in this year. They said they're fascinated by it because the cells don't age like humans. So it's like, what went wrong? And see, the difference between I believe in intelligent design and I believe in creation, but creation roughly says, we're intelligent design, but also cursed. But if you can accept that we're cursed and we're not quite working as well as we could, then we can see that there are things that are, like you said, the DNA optimized, mathematically possible. And the biophysicists are seeing this, that certain dimensions where life is optimized to the limit of physics. A real simple example is just getting your eyes to focus. If the eyes are built wrong, the light wouldn't focus right. It's built to the optimum. But then there are aspects that are, it's like, oh yeah, we can, even a human being can build like focusing mechanisms and we can get that right. But there are parts of life that are so extreme in the way that their ability to sense things like magnetic fields or electric fields, or count molecules or count photons, that are the limit.
Yeah.
That we can't engineer. There's one system in a bird that can do quantum spin chemistry to sense magnetic fields. We're not able to build any machine like that. And I sometimes even ask biochemists, I'll show them a picture of a helicase. I said, do you think you could build that? Do you think any biochemist could build that from scratch if you didn't have a blueprint? We'll say no way.
Yeah. Your point you're making on you were touching briefly on some of the factors, in the quantum biology realm that has really just started to be even truly acknowledged. Not that people had theories and, hypotheses about different things for, 50, 60 years on some of the aspects of quantum biology. But really it's been, I would say, the last decade where it's really started to actually gain acceptance. And, those things like the quantum spin for those bird navigate. I mean, I remember I read a book and then several papers on that whole thing. And my mind was absolutely blown when you really start to contemplate that. And then one of the ones that really got me, there was some bird in, it's a Scandinavian bird. And that sucker, it has the same thing, but it stops at the equator. And I'm like, okay, we're not just talking about north and south here. Somehow this thing is being able to use quantum reading, if you will, to go to a specific location that is not necessarily, yes, directional, but there's an additional component that's taking them to a specific point halfway down. You're not even going all the way to the other poles, right? Or all the way down. And
exactly.
I was thinking about that, like, wow. And then they've started to discover the exact same premise and technology, if you will, exists in butterflies and all sorts of other organisms. And it's really starting to open their eyes to, and in my opinion, it should open their eyes to recognize that from your own biological or evolutionary timeline, quantum tech came into existence undirected at the early stages of your own timeline. Not, hey, over billions of years, this, yes, it's complicated, but it's been billions of years, therefore it could occur. Like, no. This stuff is popping up per your argument billions of years ago, not billions of years later. And they don't want to address that. But now, Sal, I've been really enjoying being on for 2 hours. I know you probably have things you need to do. And
I'm kind of worn out. That's why I have big bags under my eyes.
Well, thank you so much for your time and for sharing your knowledge with us. Hope you'll come back and we can do this again and talk about more other subjects and dive into it.
Yeah, maybe we could talk offline for a few minutes afterward and about that. I'd like to thank all of our viewers live and all the ones that are watching recorded. We appreciate your time. Take care. God bless. Yeah.
Later.